Okay, let's go. I mean, let's stay here. I want to start today's lecture by taking you where we were last week when I was showing you example data that didn't exist. But I found it again. So this is one of those experiments. I don't know if you recall, but there was this. We were doing this laminar Fmri, and we were trying to find these ocular dominance columns in V1. And so we did this experiment with really, really tiny voxels, and sometimes stimulated the left eye and sometimes stimulated the right eye to be able to find these columns that are responsible for just the left eye's visual input or the right eye's visual input. Right? And when I did that, I wasn't able to show anything, but apparently it works in a different browser. So if you go to this website that's also linked in the PDF for that lecture, you can actually see this. And so let me just highlight what you see here, right? So I can go to the shape of this brain, the way that it looks inside the skull, right? So this is the way that it looks more or less normally. And what I can also do is go across depth. So now I'm in the middle of the cortex, right? So I'm between the white matter, gray matter boundary and the peel surface, but I can actually go to the peel surface, right? So it just expanded towards the outside of the gray matter, but I can also go in, and so this is the boundary of the white matter and the gray matter. And as you see, we're sampling different information going from the inside to the outside. Now, the fun thing about this type of software is that you can also just inflate things. And the idea here is that you see this sort of banding pattern more clearly in V1 than you can in, for example, V2. So let me just see, pivot. So what you see here is these ocular dominance columns. So what you see is that the saturation of the colors is much stronger in V1 than it is outside. And outside is much more speckly. And here you actually see these, these different, you actually see the columns here, right? And the idea that it's a column, of course, if it's a column, it should be more or less the same pattern on the outside and the inside, right? Because the column actually extends all the way through the cortical depth. And you see that these patterns are more or less stable as you do so, right? So much more than outside of V1. So just as an example, right? So this is more or less pushing the envelope in terms of precision of voxel size. But this is the type of thing that we can do, just as a proof of concept to show you how this works. Yeah, so I invite you to have a look at this yourself. You can play around with this and just fiddle around with the visualization and all of that. You can like flatten these out so that you can look at it. Man, I should learn how to do this. Yeah, so here you see. Yeah, so there's lots of things that you can do. I guess that point is clear, right? En zo, ja, het zijn inderdaad meer trechters. It's hard to hear me. Well, that's not supposed to happen. Let me check whether the sound is okay and what it's using. Oh, only when I was far away. Okay, I'll just sit. Okay, excellent. I'll stay close. So what are we looking at here? What are these values even, right? So what, red, blue, right? What are they? And that is really a crucial question, right? You've all seen these pictures of blobby brains in the newspaper, for example, right? And when you read this and you say, this is where the love area is, or this is where the fear area is, or whatever, you see this blobby red brain thing, right? Now, I've already told you that we're not looking at neural activations when we do fMRI, right? So we're not looking at the neurons firing or anything directly related to that, just distantly related to that. And also these colors, what do they even mean, right? Normally you would say, oh, activation. Well, it's not neural activation. And what I'm going to tell you about today, which is the general linear model approach of analyzing these types of data, it really shows you that what we are looking at in general when people show blobby brains, blobs in brains, that is the outcome of statistics, right? So what you're looking at always is a statistical value. Yeah? And it's really important to understand where these statistical values come from in order to be able to validly interpret what's going on, right? Because in many cases, when people just show you a blob in a brain, they're just peddling bullshit. So here's another example, and here I'm actually showing you statistics of a somewhat larger set of voxels. So you see, can't really zoom apparently, but yeah. So you can zoom in, and this is just to highlight. We're looking at a statistic. In this case, it's r squared, which is exactly what you're already quantifying in the practical, right? You're quantifying the ratio of explained variance. That's this, and so you see exactly what you could expect if you do something like that. Right now, in the practical, you're doing this on a single voxel, but imagine you're doing it on all voxels, and then we're projecting it onto the surface of the brain, and we can at the end of the course, you'll be at the level where you can actually do this sort of surface-based visualization yourself on a flat map. And you could set it up so that you can make a beautiful one in the browser, but we won't necessarily go there immediately. Yeah, so this is just to give you the idea that what we are looking at when we're looking at brain activations is always statistics, right? It's the outcome of an analysis that I'm going to introduce to you today, yeah, and that you'll likely have already seen happen in the YouTube videos, and if you've gone through the practical, you have some experience. Yeah. Two sort of practical things. So there was a comment about the scheduling of the exam, right? That it's very shittily like programmed right after, I think, another exam. And so there is the possibility that there is that we reschedule the exam to the Friday morning 8:30 in that same week. So I've been given that option. And so I'm going to set up a little vote on Canvas, right? So that you can vote. I'm going to answer them with their proposal tomorrow morning. So I'm just going to count votes tomorrow morning and tell them yes or no for the rescheduling of the exam. Yes or no? Well, you'll put that on Canvas. Yeah. So that's the first practical thing. The other is that this morning we had practical, and it was only online because of the Corona rules. They didn't want practicals face-to-face. Before, the rules are changing, so we now have a room for the Wednesday practicals. So the next two practicals will be in a specific room. I can look it up, but I'll post an announcement on Canvas, and you will be able to immediately also see it in the roaster.vu.nl website. Yeah. Great. yeah, I won't stray too far from my laptop. Yeah, so Florin, yeah, that was the only option I was given. So if you want to tell me when that other exam is, then yeah. I'm still working on whether it's going to be proctored or on campus. Good question. Excellent point. Okay, so that's quite distant from 8:30. Yes. Any questions about all of this practical stuff? Any questions about the brain viewer? You're very, very docile today. Okay. So I'm just going to recap a bit about the things that we talked about last week, and then I'm going to show you what the basics of an fMRI analysis are like. It's very easy, and if we end up going faster than I anticipated, I can start talking a bit about pre-processing, because that's an elaborate topic also. So when we do fMRI, right, we use that scanner that we can also use to do anatomical images with to study brain function, right? And we're using these T2 star contrast images. And the idea is that, in essence, our functional data is just a sequence of anatomical images. It's just that those anatomical images, the values in it also depend on the blood oxygenation, right? We have these bold signals that are parts of that anatomical image. So if we lower the spatial resolution and then start acquiring these things over time, very rapidly or moderately rapidly, what we get is a sequence of 3D images, these 4D data that I've also talked about a bit in the practical. And we can relate these T2 star values as they change over time to the things that we do in our experiment. That is the basic idea of an fMRI analysis. And so then the question is, okay, how do you do this, right? Drawing this relation between the T2 star values and what we do in the experiment. We're really going to use our knowledge of what happened when in the experiment to try to understand the time courses that we measure in the fMRI. That's our goal. So we will have this sequence of 3D images, and we look at a specific voxel, but we record some signal over time, right? That's the essence of having a 4D data set, is to have 3D boxes of these 3D images with their voxels, and we record them over a prolonged period of time, one sample, for example, every second. And what you get is, for example, in this experiment, there was something on the screen 8 times during the experiment, and you can see from the data when this happened. Now, these data are from an experiment that you will, some of you will be analyzing in a later project. We can do exquisite analyses on the exact timing of these activations, increases in bold signal, and thereby find out exactly what is happening inside that voxel. But this is just to illustrate that what we are working with is really these changes in the oxygenation of the blood over time. Yeah? And so this temporal component is what we have to work with. But just to reiterate, like I showed you last time, right, what we're working with is just the blood. And I think this is always really important that you deeply understand this idea of working with blood, right? I sound like a hematologist or something, but the signals that we measure are blood oxygenation level dependent, right? So we're looking at the ratio of the oxygenated and the deoxygenated hemoglobin in the blood. And this is the thing that is so important to realize, right? It's an indirect measure of the neuronal activations that we would be interested in specifically, right? Those are the things that we're interested in, but we have only an indirect measure, right? So think about this. We have time to work with, right? We have an extended signal over time. And we're going to be using our knowledge of the fact that we're looking at only this metabolic result of the neuronal activations. So we're going to put those together in an analysis. Yeah. So just to extend that caveat a bit, that link between neural firing and a BOLD is very indirect. And it would be an entire course to go into like exactly the innards of it. Now, I'm just going to reiterate this because you can imagine I find it important. So T2 star values should decrease if blood deoxygenates, right? But because the vasculature sort of overreacts, right? This changes the content of the blood in terms of its oxygenation in the direction opposite to what we would initially expect. So you get this oversupply of new and oxygenated blood to the tissue. Now, this would be a shit show if we didn't have things to work with, right? So it turns out that even though this entire sequence of very complicated and metabolic events that go through different layers of tissue to reach this vasculature and then change the blood supply and that sort of thing, we do get this stereotypical change of that T2 star value that we measure over time, right? And I already highlighted this, but I want to go a bit more in detail into the stereotypical shape of the BOLD signals that we measure, because that stereotypical shape is really what we're going to latch onto in our analysis. The fact that this response is, even though it's really complicated or the result of a very complicated cascade of biological events, it is more or less stereotypical. And that's this thing that we're going to use. And we call this the hemodynamic response function. You've seen it already. It's something like this. Okay? So again, to reiterate, this peaking somewhere between 3 and 6 seconds means we can't do anything really fast unless we want to really do all sorts of weird analyses. And if you measure very carefully, you can still find evidence of an event having happened at time 0 at time around 40 seconds. Right, so this whole metabolic thing is slow. Now, I want to go through different phases of this response, just to, because so many of them are there very consistently. A couple of them are very, very clear. Some are less, but each of them stands for a different part of this cascade of metabolic responses. Yes, the stimulus happens at time 0. Yeah. So the first thing that happens, and is what you see here is within 1 or 2 seconds, you can find an initial dip in the BOLD signal. Okay? So in this T2 star value. Now, it's tiny. As you see, it's relative to the rest of the BOLD response is not that much. And it is thought to reflect this thing that we thought we were going to measure in the first place. That is neural activity depleting the oxygen from the hemoglobin in the blood, thereby causing a negative response because deoxyhemoglobin is paramagnetic, right? That's this initial thing. It actually happens, the thing that we were going to find, we thought, right? And actually, there's a nice podcast about from the where the inventors or the discoverers of this BOLD response are being interviewed, and they recount the story of how they were like, you know, at night at the scanner, and then they were trying to set this all up. And they actually recount how they were expecting a negative response. So when they found this positive response, they were completely like, what, right? What's wrong, right? And it turned out that it was the opposite direction. This happens a lot. But in essence, if you do a very careful measurement of this BOLD response, you are able to find exactly that process that we thought and that they thought they were going to find, right? The thing is, though, it is extremely difficult to measure, right? And that's because it's tiny. You need to be extremely specific in terms of this, like in terms of voxel size. You need to sample really, really quickly, right? So if you have a 2-second TR, so you measure an anatomical image every 2 seconds, this is going to get lost immediately. Yeah. So it's really, really hard to measure. And some people even doubt that you can even measure it. So not a trivial thing at all. But it is the first thing that happens after you show a stimulus, right? first thing happens. So the neurons start firing, and this happens. But then, of course, the vasculature gets going. The blood vessels open up, and they cause this enormous increase in oxygenated blood to arrive at the tissue locally. Yeah? And what you then get is this enormous bulk, enormous peak in terms of this T2 star signal. And that is the main signal that we work with in terms of the analyses that also today we'll be looking at. So this main bold response is really the fodder for most of the analyses. Then it turns out that in order to really capture signals, we need to also accommodate this post-stimulus undershoot. So this reaction of the vasculature is actually pretty complex, and over time you get this undershoot. The idea is that you get this reduced blood flow that's even slower than this increased blood flow. So we also get this negative part. Yeah, we can estimate this shape. This is a lot of signal variation, and this stands for a lot of signal variation. So those are the two main parts of our standard bold response. Yeah.
So can the neuron be activated before the end of the relaxation?
Sorry, before?
Before this 40 seconds period.
Yeah. What? So you have a neuron that fires here. You're asking whether it can fire anywhere in this interval also.
Yeah. For example, after the 3 seconds after the source signal, if there is another signal, will there be an interference or? Yes,
very good point. So that is really the essence of the analysis that I'll be talking about today. Yes, and this paper that you're.
Sorry, can you repeat the question?
Oh, yes, of course. I keep forgetting that. So the idea is if another event happens, let's say somewhere here at 7 seconds or something like that, what will happen to the bold responses, right? Can that even happen? And what will happen to the thing that we end up measuring? Yeah. So if we have different events that are separated, let's say 10 seconds in time. Yeah, we're reading this paper for Friday, right? That's about whether the link between neural firing and the bold response constitutes a linear system. Yeah, linear time invariant system is the technical term, and that actually, if you can validly assume that what you're looking at is a linear time invariant signal or system, that means that if you have an event at time 0 and you have an event at time 10, for example, all you need to do is just add the responses to those two events. Nothing more, just adding them. And that simple fact of being able to add these things together and then pretend like nothing more complicated is going on, this linear systems assumption is what underpins this general linear model approach that we'll be talking about today. And I'll be going in a bit more detail later on about why this is the case and what types of signatures do you expect or don't you expect. Yeah, this is a really good question. It's really fundamental that we can actually assume this, okay? Because if not, then we need to actually take into account in our possible analyses all of these possible interactions between all the different events, right? And that would be a terrible thing to have to do, right? That would complicate our analyses so much that it would become more or less intractable. Yeah. So yeah, good question. So again, to reiterate, right? I think this is something that that people can become sort of enamored with the fact that we have beautiful images in fMRI, and then they forget that certain things you just shouldn't try, right? So there's a lot of people that try to do too fast an experiment using fMRI, and then they end up with more or less nothing, right? So it's important to realize if you want to do a fast experiment, you should use EEG or MEG, right? It's also important to realize that I'm showing you this sort of stereotypical time course. Yeah? And this stereotypical time course is usually assumed to be exactly the same or close to exactly the same across regions of the brain. I have a list here. Across subjects, across age, across brain regions, and across things like cognition, like different types of attention, for example, right? So this is thought to be, or assumed to be, identical for everyone, every brain region, regardless of whatever. And you can already feel where this is going, right? That's not the case. Yeah. So it is a very helpful assumption because, you know, if there's a more or less stereotypical shape and people don't differ so much between their bold response shapes, then we can sort of use it, right? But it's not perfect. And so I'm going to talk also a bit about how we can improve on this, right? And how can we step away from this assumption that everybody has exactly the same bold response. Because you can imagine that if you can actually take different bold response shapes into account, then you can actually perform a better analysis. Yeah?
Can you explain how this bold signal is related to the action potential? Measure EG at the same time.
Uh-huh.
How is it related?
Yeah, so it's related in a very vague sense, right? So it's more or less an open question how it's related to neural firing. But we know that a large part of the bold response maps more or less linearly to firing rate. Yeah. So in one of the previous lectures, there was a slide on we had a stimulus that was changing in contrast with different contrasts, right? And you get stronger firing if you increase the contrast of the stimulus. It becomes a stronger stimulus, so you get stronger firing. And that relation from like a dim stimulus to a strong stimulus, getting more firing rate, you actually see that also in the bold response. But there are more things to it than that, right? And so that's one of those things where, again, we assume that these things are all linearly related, right? And if you assume it, then you could do statements like, I'm analyzing this thing with a GLM, and then my neural activity is now this, right? But that is a chain of reasoning that goes across many different assumptions, right? And so that's also something of a general point here. We're leveraging all of these assumptions, right? And to the extent that they hold, that they're reasonable, they're valid assumptions, this makes sense, right? Because then your inference is correct. But there are these differences, right? There is a decoupling between firing rate and the bold response that you measure, right? So there's a hole there in our assumption, right? And if we're not aware that this hole exists, we are going to draw conclusions that are going to be flawed in the end, right? And similar things hold for the shape of the bold response, right? So we have all these different terribly complicated machine, all these different cogs working together to produce something that we can try to interpret. But we need to be aware of the fact that all of these different levels of analysis hold their own assumptions. And like everything, like these assumptions are limited, right? At some point they will break. And so I want to show you an example of how the shape of the bold response differs, because there's one specific aspect that we know differs, that we know this bold response shape differs across. And that's something that we've already looked at also at the beginning of this lecture. That's across cortical depth, right? So I've already talked to you about, we've spoken a bit about these superficial layers having larger veins, right? And that the blood starts at the bottom near the white matter and then flows up, right? So the average diameter of blood vessels here is larger than here. And this has an effect on the shape of the bold response that I want to highlight. So in this figure, what you see is these bold response shapes fit for single voxels. So if you take the single voxel from a large vessel, right? So that's not even in here, but that's somewhere at the top where a lot of blood is pooling. What you get then is you get a bold response that starts slowly, that rises very, very far, because there's a lot of difference between oxygenated and deoxygenated blood in these large vessels. Here it's normalized to 1, but they are much larger. I'll show you that in a bit. It peaks later, and then it's much broader also. And this makes sense, right? If this is the latest stage of this entire accumulation of blood, then of course it's going to be a wider wave, right? So you can really think of this sort of thing as a wave traveling through the tissue, right? And then if you look at the deep gray matter, that's the start of the wave, right? That's where you measure the wave more earlier on, it starts to rise earlier, it peaks earlier, and it also descends earlier, right? So that's the first part of the wave. And then that wave becomes broader as you go to the deep gray matter. You see that it
Sorry, can I
becomes
ask a
broader.
question?
Yes, of course.
Yeah, can you explain again about the large vessel? Why it's like more spread out or something you said?
Yeah, so think of this as let me just quickly think of a metaphor. So let's say it starts raining somewhere, right? When it rains, it starts to pool, this rain starts to pool into little streams, right? And they flow into larger rivers, and at some point they end up in the sea, right? I don't know if this metaphor is going to work at all, by the way. But what you expect, of course, is that there's going to be a delay. If you look in a stream, you're going to be very quick in measuring the rise of the level of water, right? And you can also imagine that when it stops raining, that increase in the level of water will also go down pretty quickly, right? Whereas if you go looking into like the delta of a river, it will be later, right? Because it needs a bit of stream to get there, and it will also sort of diffuse out. So it's going to be a longer rise in the water level. Does that sound like an explanation?
Makes sense.
Yeah, so think of this large vessel as you're in Rotterdam, and it's been raining for a while somewhere in Germany, and you're looking at the level of the Rhine sort of thing. Yeah?
Okay, great, thanks.
Yeah? Okay,
Yes.
so yeah, these analogies, sometimes they work, sometimes they go horribly wrong.
So first they go from that via, then they go to the end.
No, so sorry. So the question is about the direction of blood flow through these layers. So what happens is the blood. So the arteries also run at the top. But we're not interested in that right now. Then they go down and they more or less perfuse everywhere. So they go in everywhere with tiny arterioles, more or less everywhere around the gray matter, right? But we are sensitive specifically to venous blood, so we don't necessarily care about the arteries. Yeah, so that's something that I haven't talked about, but we're focusing specifically on the venous side. You can do different things with the scanner and then focus specifically on the arterial side of things, but that I don't think we're not going to care about that during this course. No, we are only looking at veins. So not arteries. Yeah. So, oh yes, it does. Definitely, yes. But the way that we're measuring it, so this is one of those intricacies of how the scanner picks up signals that have to do with exactly how protons, blah, this, blah, that. We are specifically looking at the venous side of the vasculature. Yeah, so the arterial side is something that we are in general not looking at. Well, so as blood goes from the arteries to the veins, some of it is used, right? But normally, like in a sort of a rest situation, it would just be, let's say, 60% oxygenation in the arterial side and 40% in the venous side, right? There's a drop, but not that much, right? So there is still oxygen at the venous side. Yeah. So it's not an all or nothing transition. It's very gradual. Yeah. But the point is, yeah. So and I think maybe I can grab a hold of that rain analogy, right? So in the way that we are measuring things, we're only measuring it after the water hits the ground, the rain, right? And we don't care about the whole supply side. So the clouds, we're not measuring. Yeah. I'm now going to stop with this analogy and continue. So if you do this, so this is just a single set of voxels, right? These are single voxels, and you can actually measure the bold response in a single voxel. Then you can also pool across different voxels, and you can get a better idea of, okay, how do these things change? You definitely see that you get a time to peak earlier, further away from the peel surface, and a smaller response and you get a larger response at the outside of the brain. Yeah? This has all sorts of repercussions, but I just want to highlight the fact that this is within an individual, right? And the distance between these different curves is millimeters away from one another, right? And so even in that close of vicinity, you get pretty strong changes in the shape of the bold response. Okay? And this just goes to illustrate the fact that we can use this sort of general shape. Yeah, they look sort of similar, right? But they are importantly different. Yeah, so we can use this assumption of a stable shape, but if we do, we need to always realize that this is just an assumption. Any questions? Yeah, blood drain. Is this point clear? So assumptions are useful, right? But they are limited. And this, we'll see this happen over and over during the course.
Sorry, there was a question in the chat about the drainage and the supply. So like, is it only one like unidirectional, or is it bidirectional? For example, if I think about feedback connections between layers, then does it have anything to do with the blood supply direction, or is it completely independent and it just uses up the oxygenated blood in either way?
Yeah, so the neurons don't care about the blood supply necessarily, so these are decoupled. But if you were to expect some activation only in the middle layer, for example, then you expect the bold responses there specifically to increase. But the problem is they won't increase just there, because the blood that gets used at layer 4 will actually then be drained upward. Right, so if you have neural activity in the middle, in layer 4, let's say, you will actually be measuring that in the MR signal, not just at layer 4, but also in the upper layers, right? So because of this draining. And there you have a big problem of this laminar fMRI. Yeah, but important parts of the answer are, no, there is no direct link between the neuronal activations and the layers, right, and the bold response. The bold response because of this drainage, it smears the bold response, even if it does happen in a specific layer of the cortex. Yeah.
Okay. Yeah. Thank you.
When you talk about layers here, are you talking about the layers of the scan, or about like the layers of the different layers of the brain? Or
Yeah.
is it...
So layers of the brain. Yes. Yes. That's a good question. So these exist regardless of how you scan them, of course. Yeah. Okay. So how do we measure bold signals, right? So we've talked a bit about the bold signal, and I've talked a bit about like things like Teslas and field strength and things like that. But there's a lot of things that you can let the scanner do, right? You play a different program on the scanner, and it will perform just a different type of scan, right? It will do an anatomy, it will do functional images. But even if you're focused on functional images, you do. The thing that we're looking at is only aimed at veins, right? And then we have other sequences that look more at arteries. There's a whole different, any program that you play will have a different type of data associated with it, and different caveats, right? And different assumptions that you need. But in the basics, we're still using the scanner, right? We're getting these 3D images once every certain time, and then we repeat that process, right? So if we are in a regime where we do that, we have specific things that we can work with. And so a couple of concepts that are important to understand when you're reading the literature, for example, are TR, right? So that's the repetition time. And we can vary that, right? We can dial this at the scanner. We can make it really slow or really fast, right? But it's part of a trade-off. The longer you sample signals from these magnetic moments in the brain, the more signal you get, right? So if you want really good signal, you want to sample really long. Yeah? The problem is we're doing functional MRI, we also want time, right? So we can't just accumulate these signals really, really slowly, right? And then there's another trade-off, which is that we can also change the voxel size. We can go to 250 microns if necessary, so tiny, tiny voxels, or we can make 5 millimeter voxels. But there, the trade-off actually goes in the opposite direction, and it also interacts with this repetition time. the smaller your voxels become, the less signal you get. Right? So you can want to be really, really precise, but you're never going to be really fast, and you're never going to have really good signals, right? Because tiny voxels are always going to be low SNR signals, or low signal voxels, and you're going to need time to get those single values. Yeah, so you're not going to have time resolution there. Okay? So this also means that you need to really think when you're doing an fMRI experiment. Everything hinges on the choices you make here. Am I going to do 2 millimeter voxels or 1 millimeter voxels? Well, there's an 8-fold difference in terms of the volume. So, 1 millimeter voxels are going to be terrible in terms of the signals that you get. Right? So are you willing to pay that price? And so these are just a couple of the dimensions that you need to think about. And in terms of MR physics, there's a lot more that you can vary. And all of those different parameters have their effect on how good your data are. Okay? So you need to be, you always have to work together with the people that are responsible for the scanner to get the best results in your specific experiment. Yeah?
How precise it is possible to the information from the most precise resolution to any?
I'm not sure whether I understand your question. So we can measure up to 250 microns, at least in a single direction.
And do this each every 100 milliseconds, and after that, put any needed.
Yeah, you mean like resample it and just say I'm going to average 100 voxels together so that the signal is going to be better. Yeah, so some labs do that, but yeah. In some regimes this works. In many regimes, you just get your signal just collapses if your voxel size becomes too small, and you can't even look, like if you look at the images that you get out, it's just shit, right? So at some point the whole thing breaks down, and then you can acquire more data and average, but you're not going to have anything to work with in the first place, right? So there are regimes where this works. But they are limited and they're not used that much. Yeah, so yeah, good point. But no. Yeah. Cool. So let's say you want to be really specific. That means that you can make the choice of saying, do I need whole brain? I can also just focus on a specific part, right? And that means that I can leave out 90% of the brain, which gives me a 10-fold increase in what I can do in that portion of the brain that I'm interested in. So you can do that. It's just very problematic if you then, there's two bad things that can happen. One is you need to target where you're scanning really precisely, because if you miss it, too bad, right? And the other is you can have a beautiful result from this 10% of the brain, but then you might think, but what about that other part of the brain? Right? You're fucked, because there is no other part of the brain. You just zoomed into that cubic centimeter or whatever it was, and you have no view on it, right? And it's happened to me in practice, where I zoomed into a portion of the brain, I got this beautiful result, and I was like, but what about, right? And then I'd already blown thousands of euros on scanning 20 people and being really spent a lot of time on it, and I couldn't publish it the way that I wanted it because of this. So yeah. So we talked a bit about magnetic field strength, right? You can get a better scanner with more T, right? And then in principle, you can get better signals. Okay. Now, one thing that we generally do when we record fMRI is we use echoplanar imaging, which in most cases means that we measure slices, right? And what that means is also that you need to be aware that every slice of the brain, let's say you measure like so, but every slice in the brain was measured at a slightly different time. Okay? And this has all sorts of repercussions for our analyses also. So we need to know where our data came from in order to be able to analyze them well. Yeah, and then in the next lecture, we're going to go into the enormous amounts of pre-processing that we have to do to make sure that when we analyze a given voxel, we're actually, we know where it is, for example. Yeah, so it's like a typewriter sort of thing, right? You cannot measure the whole brain at once. Like a typewriter, you have to write one line at a time and then go to the next line. And so that's, yeah, so we can only acquire one slice at a time when we do this standard EPI paradigm. And so you measure one 2D image, and then you zoom up, and then you measure another 2D image, you go up, another 2D image.
Time zero, we don't have.
Yes, you only have the bottom slice. And then at time 50 milliseconds, you have the second. And then at time 100 milliseconds, you have the third. And then you count the slices until you're at the top of the head. You're at 2 seconds or so, and then you start again. So every slice, at every slice, there is a 2-second difference between subsequent time points. But they all happen at different times, these different slices. So you're really zooming through the brain. And then when you're in the scanner, I don't know, some of you had been a subject, right? When you're in the scanner, you actually hear this. You can hear the scanner going, and that's when it selects the different slices and is zooming through your brain. You don't feel it, but that's what it's doing. Yeah. Okay.
Is this the reason for a trade-off between signal and voxel size?
Yeah, so if you have larger voxels, these slices are thicker, right? And so you don't need as many slices to go through the entire brain, and so you can scan faster, right? And the same type of logic also holds within the slice, right? Because you need to acquire less pixels inside that single slice. And so you can also be faster, right? Yeah, so that's a large part of that trade-off between speed and voxel size is exactly that, sampling. Right? But it's more complicated than that because MR physics is a zoo of, incredibly complicated stuff. So I can only scratch the surface, I'm afraid.
So when we see images of like the brain and captivation, we're not actually looking at what's in the brain that's specific.
No.
We're looking at.
Yeah. So see how there's like all of these different, aspects to our data that we need to understand before we can actually interpret an image of a brain with an activation in there, right? So what I want to do now is have a brief break for your sake. And then we can talk about the major analysis strategy that we have to actually take all these different assumptions and produce a valid analysis. Yeah. Okay, let's restart because there's only so much time. I have so much to teach you. I have to go back there, otherwise they won't hear me. So, who of you have ever fit a general linear model? If you are in AI, you'll be, of course. Okay. Good. Then you have a clear idea of what this is doing, and it also means that at least the basics of the notebooks and the practicals should be easy. If not, I'm going to try to give a conceptual introduction that I hope will help you finish the practicals. So, what I'm going to introduce is this general linear model approach to fMRI analysis. And the idea here is that we try to relate the bold responses that we measure to a expectation that we have of what a signal would look like. And we're going to construct this expectation using our hemodynamic response function. So an important aspect of this type of analysis is that it's model-based, right? It's this M stands for model, and it's not for nothing. This really is you're modeling a response. And again, like I said before, you're using these assumptions, right? And then the question becomes, how do we create these expectations that we're going to compare to our bold responses? That we're going to try to relate to our bold responses. And for that, we are going to use this idea of this linear time invariant system, right? This idea that we can just sum up responses to imagined events using this hemodynamic response function, and it will be okay. So the idea here is that if we were to have a single event that happened at time point around 0, this would be the bold response shape that we would measure. Yeah, so we just talked about that. There's this quasi-stereotypical shape of the bold response that we record. There are differences, like I highlighted, but grosso modo, you get a response similar to this. Now, this linear time invariant assumption then allows us to say, but what if I don't have a single event, but if I have many and they are ordered such and such over time? And imagine that, of course, I made the experiment, right? So I controlled what happened here, right? Now, this linear systems approach allows us to say, well, if I have this response to a single event, I can perform a convolution operation, and I'll explain what that is in a bit, to get this expected bold response. Okay? And so where this had a peak at 1 second and this had a peak at 5 seconds, so there's this delay between these, right? There's also a delay between these of exactly that difference in time, right? And where this one is very peaked at a single second, this one is very wide. And what you see here is also, these are very peaked and this is very wide, right? So there's, this is the result of convolving something with a smoothing filter, which is what the hemodynamic response function is. And what is this convolution? I just took this little video from, so you all know a convolutional neural network, right? So there, the same thing happens across an image, right? But here I'm focusing on what happens in just the time domain. Okay? So here you have two functions that I'm convolving with one another, right? And what you see is, in essence, what you do is you just take the multiplication of the two with a certain difference in time, and then sum that up over time. So it's a convolutional integral. You see it starts rising. The overlap of the two signals is maximal when the left side of the red thing is here, and then it starts to decrease. And you can imagine that if this is your neural activity that you're expecting, and this is your bold response, then this would be something akin to getting a bold response like that, and it would smooth everything out, right? And if you have a very complicated experimental thing, then you get a very complex shape of a time course that you expect, right? So if you have some step function, for example, and you have the hemodynamic response function, this is what you get out. So it looks a bit similar to this one, but it's prolonged in time. And what this linear time invariant property is that this paper that you're reading for Friday also is about, is that you can do this thing for any sort of activation that you expect. So you have some expectation of what the neurons are doing. It allows you to create a very specific expected response for what the bold response should be doing. Yeah. I want to go back to this idea. So this is how we construct expectations for our different time courses. I want to now go back into this idea where we're representing this HRF, the standard HRF, using a fixed shape. And we just already spoke about how this is not really true, right? We know that this shape differs even at millimeters distance. So what can we do to not have this assumption be such a problem? Now we want to sometimes also fit this shape. And so that means that we have to describe the shape. And so what we can do is describe the shape of the bold response in different ways. And one of the projects will actually go into detail on how you could do this. So one of the projects that you end up writing a project report about is actually about describing the hemodynamic response function shape. So what you can do is try to estimate the shape of the bold response by tallying a single number post-event, right? So you just, what this analysis in the end ends up doing is you say, I want 0 times that, 1 times that, 3 times that, 5 times that, and so forth and so forth. So you get a bold response shape like this. And we call this a finite impulse response model. I'm not going into the details for this type of analysis here. I just want you to understand that when people talk about a finite impulse response model that they're fitting, it's a specific type of GLM model. And what they're interested in when they're doing that is to estimate the shape of the bold response in detail. Okay? You can also separate a shape, not in terms of its unit time points, but also in terms of its frequencies. And then you use a Fourier decomposition impulse response fitting procedure. And you can imagine that if you use only a couple of these, you get a very smoothly varying bold response. And if you get more into higher frequencies, you can have more quickly varying shapes of the bold response. So people use this type of approach if they're interested in fitting the shape of the bold response. And people use that a lot. But you get a problem. The more of these you use, the harder it is to actually perform this analysis. Another way of doing this that doesn't use so many different options is to use what we call a gamma function basis. And we want to use fewer of these different HRF shapes. And we're using our knowledge of that we sort of know what the hemodynamic response function looks like. We know that it goes up, it has this bulk, and then it goes down. Right? And we can use that knowledge. So we're going to use a specific mathematical function called the gamma distribution as a backbone, and then try to estimate the shape of the bold response on the basis of this. And so, for example, we can use this sort of the standard HRF and use its time derivative. So the derivative of this function in time, you see it goes up until this point where the derivative goes to 0. And then the derivative is negative for this entire thing, which you see here, and then it goes up, right? So that's the time derivative of that hemodynamic response function shape. It turns out that if you combine these two, you can actually end up shifting the shape of the bold response, right? So that difference between the large vessel bold response and the gray matter bold response that we just showed you over depth, you could actually capture by a combination of these two, and not just a single assumed bold response. Okay? And so this is the type of thing that you would get if you were to do that sort of thing, right? So you're able to shift the shape of the bold response to the left or to the right. And that's more or less this difference that we already saw in the data also, right? So this you can see as a very simple way of getting to those different shapes. Okay? So it gives us a lot of flexibility, more flexibility, and we can more accurately capture what's happening in the signals that we record. And you can do even more things. You could add another, what's called a dispersion derivative. There's another sort of variable in this function that we could also take the derivative of, and then you get even more flexibility in terms of the shapes that you end up defining. But, so this is all just if you want to estimate the bold response, right? So you have these different ways of performing that analysis, but I haven't actually explained to you what this analysis does, right? And so I've talked a lot about the shape of the bold response and that we might want to estimate it in detail, but I haven't talked to you about how you would estimate it then, right? How does this estimation procedure work? And so I have to go into that a bit. Okay? Now, if you want to go back, you can go back to this recording. The YouTube videos give you independent sort of explanation of these same principles. And in the practicals, you get this hands-on approach where you go through the different steps of how this analysis works, right? So we're hoping that we sort of tile so that the way that we're teaching it goes from so many different angles that you get to latch onto it, regardless of where you came from. Okay?
Sorry, I have a question about the gamma function.
Yes.
So basically, are we using it so let's say we make a measure and then we can kind of account for delays by shifting it forward or backward, because we know that it would look like this depending on, like a different time step, or what exactly is the purpose of doing this, like trying to get all these different shapes? And
so you can imagine that if your assumption of the fixed hemodynamic response function shape is wrong, then your analysis is also wrong, right? It's handicapped from the get-go. So if you're able to take this into account, the fact that, for example, the shape of the bold response is different from subject to subject, and if you're actually able to use a tailored hemodynamic response function for every subject, that allows you to much better explain the signals that you have recorded.
Okay.
Does that
yeah, thank
answer
you.
your question?
Yes, very much. Thanks.
Awesome. Yeah, and so all of this, and also the things that I'll be talking about just now, they are all about, we have measured a signal, we've conducted an experiment, it has caused these time-varying signals that we've recorded in this 4D data, and now it's my task to explain the data. Yeah. So I want to be able to model where the data came from. That's our goal. Yeah. So I'll get into the bread and butter of the analysis, and then I'll explain this in detail. Yeah. This was just to highlight the fact that, we're using an assumption and it's clearly wrong in many cases. And so we want to alleviate the impact of this assumption, right? So we can tailor this bold response shape. And how to do that exactly, I'm going to show you. Yeah. So the idea here is that you're going to record these signals, right? And you've done an experiment, and for example, you'll have a couple of different types of events in your experiment. So I'm going to use very simple experiment as an example experiment and how to analyze that. And the basics of the analysis that I'm explaining now will go a lot further, right? Even far more complicated experiments you can analyze with the same approach. But I'm going to start with a very, very simple experiment. We put someone in the scanner, and we either, at some points in time, we showed them a light flashing, and in other points in time, we sounded a beep or something like a sound over the headphones in the scanner. Yeah, so it's a very simple experiment. Sometimes there's visual input, and sometimes there is auditory input. Nothing more. Yeah. But we are now going to be interested in analyzing what is a visual region of the brain and what is an auditory region of the brain. Yeah. And so for any given voxel, we want to answer the question, does this voxel respond more strongly to vision or to audition? Is that setup clear? Very simple. And of course, people do much more complicated experiments, but for the purposes of this explanation, this is as far as we go. So what we want to do, I talked a bit about we want to construct expectations of what the signal might look like, right? And then check whether the data conform to those expectations, right? We're going to compare the data to the expectations that we have of what the signal looks like. So we're going to have different expectations, right? So we're going to have different expectations for the visual part and for the auditory part. And these expectations are called by different names. They're called explanatory variables. They might be called predictors. They might be called regressors. They're all the same thing. What they are is our expectations of what the signal should look like. So in this experiment, we have visual and auditory events. The visual events happen here, 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 and so forth and so forth, right? And we have auditory events happening at this time point, this time point, this time point, and so forth and so forth, right? So what we have here, you see that even though it happened, something happened really, really briefly, we get this hemodynamic bold response. You see that there, right? So this convolution thing that I talked about just now has already happened here. This is really at the level of what we expect the signals to look like in the brain, already having used this hemodynamic response function. And we have separate types of events, so also different shaped expectations of what a signal would look like. So in an idealized situation, in an ideal brain with no noise, if I'm looking at an auditory voxel, it should respond exactly like the green time curve. Yeah? And a purely visual voxel with no noise should have this blue response over time. Yeah? Because we know when the neural event happened, we showed the stimulus at that time point. We have this idea of how to translate that neural event to a bold response time course, right? So this is what those signals should look like. Yeah? We have a very clear expectation. But you already see that if I were to change the hemodynamic response function shape, this time course would also look different, right? So that expectation would change in terms of its shape. So our goal with this entire type of analysis is to explain the signal that we measured, so that time course, that 4D time course, yeah, so these anatomical images over time, how they change over time, using some combination of these explanatory variables. Yeah? And so you can imagine that if you have a visual voxel, you get a signal like this. You have an auditory voxel, it would have a signal like this, and they're quite different, right? And for example, you could also have a multimodal voxel that actually takes in auditory information and visual information, and it would be some combination of those two expectations, right? And we want to be able to capture signals from any of these categories of voxels. Now, the way that we do that is by saying if we have two explanatory variables, we say we have an expectation time course x1 for vision, for example. We have an expectation time course x2 for audition, right? So those are just those vectors of these time courses. We have an intercept, which is like the mean of the signal. We'll get into that in the practicals. And these describe our data. So our data are some combination of this time course for vision, this time course for audition, plus an offset and noise. But how much this measured time course relates to vision and how much it relates to audition is given by these parameters A and B. And I'm interested in those, right? Because A will tell me how visual this voxel is, and B will tell me how auditory this voxel is. Are you all with me? Yeah? So Y is the data, these are the explanatory variables, and these A, B, and C are our parameters of interest. We want to find those. Right? So we need to have a procedure to give us those numbers from data. So we have our expectations as they transpire over time. We have our data. So how do we get these numbers A, B, and C? It wasn't necessarily an open question, or I'll explain how we get those. But it's really simple, right? Because these numbers are just scaling factors, and they determine whether a voxel is visual, auditory, or something like multimodal, right? So the way this is set up is really simple. It's just a linear regression problem, multiple linear regression, multiple for the different expectations that we have, and these scaling factors that we're interested in are what's called beta weights, right? So any of you that have ever done linear regression in some, what they always say, right? The many AI machine learning methods end up being linear regression. This is that, right? We're doing exactly that same type of analysis on fMRI data. Right? So what we have is Y, of course, our data, right? This is voxels by time. So we don't care about X, Y, and Z for now. We just collapse over all voxels. It becomes a 2D matrix, voxels by time. And so that's all of our data, time by space. That's what we have, right? And then we have a design matrix, which is our model specification. Yeah? That is time by regressors, right? So our regressors, for example, these auditory and visual regressors are what constitute this design matrix. What we're interested in here, what needs to be estimated here is these beta weights. Yeah? And of course, we have residuals after we do this. That is the part of the data that we cannot explain. Now, what we want is for this combination to explain as much as possible of the data, and for this to be as small as possible, right? Because that means that we have a lot of knowledge about the generative mechanism behind our data, right? If we understand fully the processes that caused our data to be shaped the way they are, then we can explain everything and we would hardly have any residual error. But you can imagine that if you have very, very noisy data, you cannot explain all the data, right? There is going to be a lot of residual variance, things that you cannot explain, right? And so in fMRI analysis, the crux of the analysis is always to try to estimate as much of the signal as you possibly can. And this is your enemy. Okay? That's what we do. Now, in order to fit this, you have a very simple set of equations. What you want to do is minimize the squared residual, right, the squared error, because that will give you the beta weights vector or matrix that optimally explains the data. Right? So that gives you the minimum squared error. Geez, this happened again. So let me replay this. Yes. So you want to minimize the squared error. This is a squared error, of course. Yeah. So this is again that same equation that I showed you on the previous slide, but now it's sort of collapsed. And we want to minimize this quantity. And the way to do that using standard linear algebra is to do this in matrix multiplication terms. I won't go into this specifically. The course is intended for psychology majors, so we're not going to go through all the linear algebra for this type of analysis. We are going to just use this as NumPy least squares. Okay? And if you're interested in this, we can have much deeper discussions about how to do this and how we would go to other types of regression and things like that. That's fine for now. What we are interested in is just to find those beta weights. So we are going to just get them by using NumPy least squares. And you'll see this in the practical notebook. Now, that's all good, right? So you get these beta weights. And what they are is they are the scaling factors for each of the regressors or expected regressors. Expected variable. I always forget these three. There were three names. We can look them up. So these beta weights are the scaling factors for each of the voxels separately that give you the best possible description in a least squares sense of the bold responses that you measured. Right. So this allows us to collapse our knowledge of what a voxel is doing into a simple vector of beta weights. Right? We don't care necessarily about the intricacies of the time course anymore. Using this assumption of the hemodynamic response function shape and using the GLM, we can actually quantify what every voxel is doing by this simple beta weight vector and work with that. So what this gives us is for every voxel, a set of parameter weights. Now, like I just said, each of these parameter weights can be multiplied by the explanatory variables, right? And thereby reconstitute the part of the signal that we understand, or y hat, as you will find it in the notebooks. Yeah. So, of course, these beta weights that we find here are then going to tell us whether a voxel is visual or auditory. Yes? Yes, we're assuming that this is all stable over time. And you can create more complicated models. Right? That's the game, of course, to create a model that best explains the data. And if you would say like, but you know, as time progresses, the same sound will start to you know, appear less strong because you're adapting over time. Well, this is not just a statement; it's a model, right? So you can actually model this. You can say, but the auditory one should be going down in amplitude over time. And then perform this analysis too. Okay? Yes, yes. Yeah. And so these beta weights are really the primary outcome of the GLM analysis as we perform it on fMRI data. And also it's going to be the start of the statistics that we would be looking at if you look at brain blobs, for example. Yeah, so blobs in the brain. Also the ocular dominance columns that I showed you, the red and yellow things that I that I made appear by pressing on the wrong button. All of those are just statistics that are in the end derived from these beta weights. So I want to quickly introduce a few of these concepts in terms of the statistics. It's all very, yeah?
Sorry. Shouldn't we limit exclusively the coefficients to be at least positive, non-negative?
Yeah, you can do that. So you can use specific, fitting routines that do this.
Is it necessary?
No, it is not. So there are. So actually, I've started making a point that you shouldn't, because in many cases we shouldn't be looking just at activations, but also deactivations in the brain can be very informative, right? And it turns out that it's just the flip side of the coin. If you sort of pay attention to the left side of space, you actually pay disattention, if you will, to the other side of space, right? And you would get a difference in the bulge response, not just because there's an increase on one side, but also because there is a concomitant decrease on the other side, right? So this decrease you would actually be seeing in terms of negative beta weights. Yeah. I hope that's clear. If not, we'll go through this in the practicals, and I can actually, on the fly, also create situations where these beta weights can be negative. Yes, the expectations are the same for all voxels. So x1 and x2 are just a function of the experiment, and you're just fitting to see these parameters a and b for every voxel separately. Yeah, and so they start out being 1 or 0. I have a bunch of questions here, which I'll read actually. So the question is whether x1 and x2 are the same for all voxels. The answer is yes, right? So because there's a function of the experiment. The second question is, are x1 and x2 just 1 or 0, depending on whether the stimulus was shown? Initially, yes. So the neural activity that we think happens would be 1 or 0. Or if you have different contrasts of a stimulus, they might be 1, 0.5, 0.2, whatever. But yes, initially, presence or absence of a stimulus is 1 or 0. And then you convolve that with this hemodynamic response function, which gives you this sort of continuous varying change of the signal that you expect. Yeah, so at that point, it's no longer just 0 or 1. It actually is this smoothly time varying expectation. Yeah. Great. So I hope that answered your question. You can let me know in the chat. So the crux of all this type of analysis is to get small residual errors. And as we progress, we'll also highlight how we can actually sometimes denoise our data, for example, if we have noise that we actually know the origin of. We'll get there when we get there, but really I want to highlight how important it is to try to explain as much of the measured signal as possible. And what that means is you want to decrease that noise, that residual error that we have. That's also the reason why you want to have the best possible data, right? You just want to have very small error. Now these betas together with this residual error really forms the basis of all statistics that we end up looking at, right? In a brain viewer or in a publication where people show blobs. The beta weights together with the residual errors are what we're focused on, what the fodder is of this analysis, right? So one basic statistic that we use a lot in this type of analysis is a T-statistic, where it's just really the beta weights divided by the standard error of their estimate. And the standard error is just the residual variance sum. So this is the residuals, right? So the y hat is the part of the signal that we can explain. The y is the original signal, right? So their difference is the part of the signal that we cannot explain, the residual time course. We square it, we sum it, and we divide it by the degrees of freedom, that is how many time points more or less. I'll explain that in a bit. And then we take the square root of that. So this is a measure of the standard error. And you can imagine that, let me just go through that, right? So let's say you have a large standard error. Okay? This means, so you have a large residuals. So your model really sucks in capturing the variations in the time course of the data that you have. What this means is this value goes up, right? It's going to be a large value, right? Regardless of this and the square root, what this is going to do is cause a very large standard error, right? That then feeds into this fraction, which decreases your T-statistic. Okay? So bad data, that is more noise that you cannot explain, causes low T-statistic values. Okay? We'll get into what that means for your all-important p-values in the next slide. For now, the degrees of freedom are given by the number of time points in your data. So that is the amount of data points that you have to do the fit on, minus p, and that's the rank of the design matrix. So with this input, this just stems from what you did as a model. And this is very easy to calculate. Also, you do this in the practical. It's just a one-liner, of course, and then you get these T-statistics from this fraction. Right? Very easy to get t-values, and often people just report t-values. So you get a brain filled with t-values for vision, for example, right? And what you will see is very high t-values for the visual system, right? And you can do the same, get a t-statistic for auditory, right? So that's the auditory beta weights in that case. And then you get a brain image of where the auditory system is. Right? So what many people like is to report a p-value. Now, just quickly, I want to go through how the p-value that people are generally very much interested in, how do we get that p-value? Well, that's relatively simple. We have a t-value, and we can actually convert that into a p-value with a t-distribution. So with a certain number of degrees of freedom, we get a shape of a t-distribution. With our t-value, we can look up what the value of that t-distribution is at that point and get a p-value. So this is our t-distribution, right? So this is our this is the t-distribution. What you get is a value of a t-value that's very much outside the zero, right? And you can get a p-value from this specific shape of a t-distribution. Now, it's important to realize that as your degrees of freedom go to infinity, well, important, I don't know if you're into this sort of thing, this t-distribution actually in the limit becomes a normal distribution, in which case the t-value just becomes the number of, it becomes very, very similar to the number of standard deviations that you're outside of the mean, right? So just to give you an intuition as to what a t-value means, you can broadly, like through the hairs of your eyes sort of thing, when you squint, you can interpret it as the amount of standard deviations that you're outside of the mean, right? So at level 3 or 4, it also already starts to become moderately significant, if you will. We'll talk a bit more about specific statistics later on in separate lectures. But I hope that this gives you an idea of how we use our knowledge of the hemodynamic response function shape to create these expectations, and how this then allows us to say whether the signal in a certain voxel aligns with our expectation of, in our example, vision or audition, and that we can quantify this using this GLM approach, and thereby also quantify things like get statistics. Right, so this sort of thing, these images and the ones that I showed you before in the brain viewer, right? They are beautiful. But again, I want to reiterate that what we're looking at when we see these beautifully colored voxels, right? We're not looking at activation, right? It's the result of not only this translation from neural activations through the Bold response to a measured signal, it is also the result of a whole pipeline of pre-processing that we haven't talked about, we'll be talking about in the next lecture. And then it's the result of all of these analyses based on the GLM that are ridden with assumptions, right? And what this means is we're looking at, of course, statistics, and this statement, lies, damn lies in statistics, it holds for this type of result, right? You have to be aware of the fact that you have this entire pipeline of steps that leads to an image like this. And as long as you don't know exactly what those steps were in the analysis pipeline, it's actually really hard to interpret what actually this means. And so if you just show people these brain images and just say, here's the activation, you're actually glossing over a lot that can be incredibly important. And it's very easy to fool people if you just show people the end result of some incredibly complicated analysis. And I've not even shown you the parts or the pre-processing that can be even more important. So that's really the takeaway for today. I hope you get an intuition for how this GLM works, that this is a parallel analysis that we perform on all voxels that gives us these beta weights for all of the voxels that then quantify how well that voxel behaved the way that we expected it to behave according to this model. So on Friday, pre-processing. After that, the lectures are still a bit in flux because my guest lecturers had shifting schedules. So I'll be updating the schedule for, like, that's in the syllabus as I learn more. Okay. For now, in 2 days, pre-processing.
I have a question.
Yes, of course.
About the about the GLM, when you were talking about, for instance, the with the beta and the error, etc. Is that handled in the practical a bit more, because it went a bit too fast for me, to be honest. Like I have to. Do you practice with that?
Practical, you mean?
Yes.
Yes. So in the nuisances practical, it's called week 4, but you know it's sort of all over the place in terms of timing. In the nuisances notebook, we will go into that more.
Okay.
And if you have specific questions, you can always ask them on the discussions. I'll try to get into those ASAP. You can always also ask me an email, and I'll be very glad to just go in detail with you.
Okay, great. Okay, thank you.
Excellent. Tell me.